Discuss about synovial fluid and synovial fluid analysis, right? So synovial fluid is basically formed from the synovium, right? Synovium covers the joint. It forms. It is all around the joint. Synovium is basically a vascularized porous connective tissue and lacks a basement membrane. To repeat, it is a vascularized porous connective tissue and lacks a basement membrane. And what is the function of synovium? Synovium mediates nutrient exchange between the blood and joint fluid, It's a porous connective tissue, but it is some kind of a barrier between the joint fluid and the blood. Okay. What are the types of cells found in the synovium? Types of cells found in the synovium? We have what is called as the type A cell. Type A is a macrophage type of cell. It is non-fixed and it is an antigen presenting ability. It presents antigen and it is located in the superficial layer. It is mainly located in the superficial layer. It is very important as macrophage function goes. It is very important in phagocytosis. Type B cell. Type B cell is a fibroblast type of cell. It is present more in the deeper layers, right? And it contains rough endoplasmic reticulum and dendritic projections, and it is the one which is responsible for synovial fluid production. It also produces other things like hyaluronic acid, 2 lubricin, 3 fibronectin, and 4 collagen, right? So type B cell or type B synoviocyte or type B cell is a fibroblast type of cell which is found in the deeper layers of the synovium, produces mainly the synovial fluid. Type C cells are intermediate cell and we don't know what their actual function is, and it may serve as a multipotent precursor to type A or type B cell. So type C is basically a precursor to type A or B. Cell is a macrophage type of cell which helps in phagocytosis. Type B cell is a cell which is fibroblast type of cell which produces synovial fluid, and type C cell is an intermediate one which we don't know what actually function is, but it is known to produce type A and type B cells. It's a precursor cell, maybe it produces type A and type B cells. So these are the 3 different types of cells that you should know when you write about synovium and synovial fluid. So next let us go to synovial fluid. What is the function of synovial fluid? Function of synovial fluid is it lubricates the articular cartilage. The second function of it is it provides nourishment, right? We learned in another video about cartilage that the cartilage does not have, it is avascular. It does not have any vascularity of its own, and therefore it needs nourishment from either the synovial fluid side or it needs nourishment from the subchondral bone side, from the bone side. And as such it does not have any blood supply. So cartilage needs nourishment from synovial fluid, needs lubrication from synovial fluid. And what is the origin of synovial fluid, right? What is the origin of synovial fluid? Synovial fluid is made from an ultrafiltrate of blood plasma. And in a normal knee, how much of synovial fluid is usually seen? Only about 2 milliliters of synovial fluid is seen, right? What are its constituents? Now let us look at its constituents. It has hyaluronin, it has lubricin, It is very important for lubrication, that is, it reduces the coefficient of friction. Next, it produces proteinase. It contains proteinase. Next, it contains collagenase. Then it contains prostaglandins. Right? Synovial fluid exhibits non-Newtonian characteristics. That means non-Newtonian characteristics. That means the viscosity of coefficient is not constant. The coefficient is not constant. The fluid is not viscous linearly. It is not uniformly viscous. It is not, coefficient of friction is not constant. It is not uniformly viscous. These are the two characteristics, and it demonstrates non-Newtonian type of characteristics. This is a very important feature of synovial fluid. Coming to synovial fluid analysis, right? In a normal when you aspirate synovial fluid in the knee, we saw that it only has about 2 milliliters in the knee, 2 milliliters of fluid. The normal knee, it is completely transparent. There is no color, it is very clear. Viscosity is high. The WBC count per millimeter is about less than 200, and among the WBCs, less than 25% are polymorphonuclear cells. In case of non-inflammatory pathology, the clarity, it is clear, it is yellow in color, the viscosity is still high, the WBC is 200 to 2000 per millimeter cube, and the polymorphonuclear cells are less than 25%. In case of inflammatory pathology, for example, like rheumatoid arthritis, it is translucent, the clarity, the color is yellow, the viscosity is low, and you might find cells of up to 2000 to 10,000 WBCs per millimeter cube. And polymorphonuclear cells are more than 50% of the total leukocyte count. In septic arthritis, it is opaque, it's dirty or yellow. The viscosity can be variable depending upon how thick the pus or discharge is, and the WBC count is more than 80,000 per millimeter cube, and the polymorphonuclear cells are more than 75% of the total leukocytes. In hemorrhagic, for example, in any kind of bleeding disorder, you will find a completely bloody aspirate. Post-trauma also you can find a bloody aspirate. The color is usually red, and the viscosity can be variable depending upon the hemorrhage that has happened. The vessel count, the blood total leukocyte count or WBC count is reasonable, 200 to 2000, and polymorphonuclear cells may be 50 to 75% of them. Depending on what you suspect, you can also get what is culture sensitivity. If septic arthritis if you suspect malignancy, you can do cytology. Crystals, this we know very well. There are two types of crystal disease. One is the CPPD or pseudo gout, and the gout. In the gout, you have negative birefrigerant crystals, negative birefrigerant. In CPPD, it is weakly positive crystals, This you have to see under the microscope, and then you can differentiate between CPPD crystals and gout crystals. So this is in brief about synovial fluid and synovial fluid analysis. You should be able to reproduce this table in your exam. It's quite commonly asked, and you should also be able to answer this question in your OSCE as well. Hopefully this helps you. Thank you very much.